As an introduction, I'm going to first talk about what the ChemAxon calculators are and how you can use them. So very often during the drug design processes, you come to a point when you want to calculate very important physicochemical properties for your molecules. And in those cases, you might ask questions like, will my lead candidates or drug molecules be ionized at blood pH, or will they have optimal lipophilicity, or will these molecules be soluble in water? And ChemAxon calculators provide solutions to these questions. So they are high-quality physicochemical predictions, mainly for the preclinical drug design. The calculators cover a wide selection of property predictions, so we can predict basic elemental analysis functions like molecular mass or isotope distribution. We can predict pKa and microspecies distribution, which is the best on the market. We can calculate lipophilic properties, so log P, log D, and hydrophil-lipophil balance number as well. A new, quietly new prediction is the solubility, water solubility prediction. We can even calculate pH-dependent solubility. We can predict NMR spectra, so carbon and hydrogen NMR as well. We can generate different set of isomers and tautomers. And hydrogen, for example, hydrogen bond donor acceptor sites and counts, and many other important topological and geometrical descriptors. All these calculations come with great versatility. So, for example, if you are a medicinal chemist, these calculations are available as one-click plugins in Marvin Sketch, for example, and you can calculate and store them, for example, in Excel, in your Excel sheets. But if you are a more advanced computational chemist, these calculations can be also used as a highly customizable tool. So they are available as a command line interface. They are all integrated and available in IGC and now in Plexus Design as well. And you can build a whole calculation workflow in KNIME or Pipeline Pilot using the ChemAxon calculator nodes. Before I go on to the new cool stuff we developed, I would like to give you a valuable reference for the calculators. This article was published last year in the Nature Reviews of Drug Discovery, and it was a joint project of 4 major pharmaceutical companies: AstraZeneca, Eli Lilly, GSK, and Pfizer. These 4 companies put together a joint set of about 800-ish compounds nominated for development during 2000 and 2010. And they try to analyze and link the attrition of these candidates to physicochemical properties. And all 4 companies used a wide set of ChemAxon calculators to do this analysis. For example, log P, log D, pK for ionization sites, molar mass, and other important predictors. Without going into too deep details, one of the main conclusions of the study was that when we want to somehow optimize or control the physicochemical properties, lipophilicity has a very important role in optimizing the drug candidates. But the overall whole conclusion was that controlling physicochemical properties in general has a very important feature in drug optimization. Our group worked hard during the last year and provided new features for the calculators. The first of them is the solubility training feature. We started to develop this feature from a benchmark. So we obtained an open experimental data set of solubility values from the Campbell database, which showed not very bad results using our basic solubility calculator. First, we tried to investigate what caused this problem, and we did an overlap analysis between our intrinsic training set of the solubility training set and this AstraZeneca set. And what turned out is that there wasn't too much overlap. So that caused the problem of the initially bad results. We cured this problem by introducing solubility, the training capability, which means that the user, if you have a very good experimental data set, you can use this training set to train your data. On this chart, or on this plot, I show the initial results before the training on a small test set. And on the right side, you can see the same result, but after the training. And the training was introduced using ECFP fingerprints, so some kind of similarity. And I highlighted here one molecule which shows that the quality of prediction after the training is very high using similarity predictions. Our future plans, we are now currently developing a new module for DMSO, the PK prediction in DMSO, which is a very, or most common polar protic solvent in chemistry and analytics. So this will be a very valuable tool for your chemist besides our water PK prediction module. And I would finally like to invite you to our demo site where you can see, I can show you our new demos for some of the calculators. Here I just show the solubility demo. So on the left side, you see a Marvin.js window, and when you draw a molecule, the log S curve automatically shows up.
Yeah, can I just ask then, with the log S calculation, you're going to have a real domain of applicability problem if you've got using similarity to do it. So do you give sort of some estimate of that in the output in terms of the probability of the prediction or the accuracy?
Yes, so currently what we do is that from the training set, we just, if we see that there is a very similar molecule, then we suppose, or we can say that it's very likely to be acceptable. But in the future, we will do, we'll somehow calculate some kind of probability or acceptability of the result as well.
I was calculating PK microspecies in KNIME using the KNIME node. And then I realized that it would also include like protomers of very weak acids and bases. And I didn't find a way how to kind of set the PK threshold. Is there a possibility to do it?
Yes, so I think so not all options are available in KNIME. So there might be some difference from the fully customizable calculations like ChemAxon's or Marvin, but I think we can work on it. So if you have the need to have more customizable calculations, then we can introduce it in KNIME as well. So
Okay, good.